Hello everyone, I'm Maria Nadestad. Welcome to the Genomics Show, and this is the long requested video on project ideas. How can you make a bioinformatics project out of thin air that you can learn from and so on? This is oriented at late undergrads probably, and early grad students who are looking for a first or early project to sink their teeth into, to learn how to run bioinformatics tools and kind of go through the whole sequence of how you might do bioinformatics. Well, I got this question a long time ago, and at some point I made a Twitter thread, which I'll link in the description below, and I'll also show something here, maybe. I asked Twitter, because luckily I am plugged into the genomics community enough that when I ask questions, smarter people sometimes answer. Other people in the genomics space jumped on this thread, helped me answer the question, and so I'm going to compile some of the wisdom into this video, but I also really encourage you to go and just read the Twitter thread yourself as well. I have taken their wisdom, I have played off of it and added some of my own, and will include some general ideas here in this video. So let's get into it. So some ideas for projects are, many of them, you start with a paper. So the first one is reproduce the results of a paper. If you don't know where to find papers, the bioRxiv is a good place to start, because it is free and open. You look for a paper with a detailed method section and data available, and you just try to reproduce the exact thing that they did. You might have to look a bit to find a paper that has the data available and a solid method section that's detailed enough that you can actually reproduce their results. Then you try to reproduce it, you might try tweaking it a little, see if the results are different. You can kind of follow your own curiosity on that and see how well their results hold up to actually, trying to reproduce it. Or another thing you can do is reproduce the paper's analysis on a different data set. This is easier to do for slightly older papers because new data sets would have come out after their publication, so then the original paper did not have the chance to do their analysis on the newer data set, but you can. And that could even, lead to a paper of your own if you're, basically seeing, like, does their analysis hold up on this new data set? Do you see anything new and interesting on the new data set? So that's also a really solid way. Now the third one is you can reproduce an older paper's analysis on, say, a newer reference genome and compare to the original results. Someone mentioned this in the Twitter thread related to gene expression analysis. So a lot of these analyses, build a lot of things on top, but the reference genome is one of the early steps of the analysis, or is used in like the alignment step, which is a super fundamental part of any analysis. So if the entire rest of the paper's work is built on top of that, changing the reference genome could change the results. And that would be interesting, and that is also worthy of a paper if you find something interesting when you're doing this. Otherwise, you just learn a lot. The fourth one is a particularly great technique, and you don't even have to go find the perfect paper. You can benchmark similar tools against each other. Like take all the variant callers, take all the differential expression analysis tools, take all the aligners, take all the tools of a certain type, right? And compare them to each other. See which one performs better. Find ways to compare them against a truth of some kind. Since you're probably pretty early on, a good way to do that is to find the papers of the tools and see how they compared against previous tools. The latest tool will usually compare against whatever are the top tools that it sees as competitors. And so you want to usually just find the latest of the papers, see what it does, and see if you can reproduce some of those benchmarks. If they are not benchmarking against all of the methods that you would like to see them benchmark against, you can do that analysis yourself. So when a new tool is published, they do benchmark against similar tools, but we should be a little skeptical of that because they might have sometimes unintentionally biased their choice of data sets or tuned their method to the data set that they're evaluating against or evaluating on. But of course, they didn't do that amount of tweaking or something for the competing tools. So it ends up being that, whenever a new tool is published, it always says it's better than the previous tools, right? It's always better than the one it's comparing against. Sometimes you will get some figures in the paper showing, oh, but we're a little worse on this or something like that. But generally, new tools have to outcompete the previous tools in order to get published. But it might be that some of those analyses are not perfect, and it might be that when you redo them, you won't see the same results. And that is always interesting, and that can be really helpful. You can always publish a blog post or something on this, even if it's not big enough for a paper, which it totally can be, especially if you find something different than in any of these previous papers that have, than the tool makers themselves have written. Benchmarking is super useful. And there are always journals that will take these types of papers, even if you didn't find anything like totally amazing. So you can absolutely do a real project on this. As a twist on this, try changing something in your benchmarking evaluation to see if the winners change. As an example, for variant college, you could downsample the coverage. So just like downsample the reads to get lower coverage to various different levels of coverage and see what happens to the accuracy of each tool. You will often see some tools that are really good, but as soon as you go below 30x coverage, they get worse, for instance. So this can happen. Especially try to do some of these things if they're not already done in the previous papers. Or if they are, you can just reproduce the results, right? So I think that's always a super interesting analysis to do. And it is something that lets you have real results, and it's something that teaches you a lot because you had to learn how to run the bioinformatics tools and how to evaluate the results, how to look into them and see, oh, why is this tool underperforming in these certain conditions? Is the lower recall because of a single type of variant? Is it more in SNPs or more in indels? There's all these different ways you can split it out and dig in further. And so this gives you just like a wealth of different analyses that you can do and so much to learn. So benchmarking would definitely be a favorite. I think this was mentioned at least 3 times in the Twitter thread. Just like benchmark different tools against each other. It's fantastic. One idea that I liked in particular is for someone who, if you're the professor listening to this right now for an undergrad class, you could, for instance, have something where each student or each group of students uses one tool and then they all combine the results at the end. So then that way you're kind of crowdsourcing this benchmarking analysis. And that can be really useful. Okay, so those were some very nice general ideas that I really liked that came out of this Twitter thread. With significant rambling from me. Okay, so there were a few other specific ideas that I liked. This is a very small subset of them. One is to look for contamination in older assemblies. This is a mistake that people keep making. You can have contamination from other species, such as bacteria that were in the lab, or even from the human's own skin cells or something that ended up in the sample. All sorts of interesting things can happen and can end up in genome assemblies. I think the assumption is that in the past this used to happen more, but you can try to see if that's actually the case. An example I really liked there is looking for Illumina primers, and in the Twitter thread, someone did link a list of Illumina primers. So if you want to go through a bunch of old assemblies and just align the primers to it and see if you get a perfect match anywhere, then you might have found an assembly that has Illumina primers inserted into it, which is probably always a mistake. Illumina primers are not supposed to, exist in nature for real. Although I'm not sure exactly how they made sure that never happens. So anyway, that could be a fun analysis to try. Another one is PacBio, or general long read gene fusion findings. So Liz Zhang from PacBio, good friend of mine, who I've worked with before, she says that long read fusion finding would be a good project, either to compare existing tools or make something better. Long read aligners still struggle with consistent fusion findings, she says, so weeding out false positives or artifacts using annotation or matching short read data would be very helpful for the field and would be a very interesting project. You could end up with the best long read gene fusion finder, and so that could be fantastic. So that's another very specific idea. Another one is one that I have actually done myself, which I thought was really fun. So when I was interviewing at Cold Spring Harbor, I interviewed with one professor named Mickey Atwell, and he gave me this idea as a getting started in bioinformatics kind of fun project. And I ended up using it for my linear algebra final project, where we're applying linear algebra to different things. This was using PCA, which is a technique that comes directly out of the eigenvector, eigenvalue analysis that we do in linear algebra, so it's kind of cool. So if you look for a paper called Genes Mirror Geography within Europe, this was taking genotyping data, SNPs I believe, from, it's been a long time since I've done this, so I will recollect to the best of my abilities, but it was using SNP data, genotypes from many people, and basically just collecting all the data, putting them in a giant matrix, and doing PCA on it. And you've probably seen that gives you a two-dimensional plot. So you're taking, many dimensional SNP data, which is just, do people have this or not? Do they have like 0, 1, or 2 copies of an A or whatever? You just change those into numbers somehow, get a matrix, and then run PCA on it, and you get a nice chart. And this can actually give you, it turns out that when you just run PCA on SNP data, you get like in Europe, you get the north-south axis roughly being the first principal component, and you get an east-west axis being the second principal component. It was so cool. I reproduced this just with HapMap data, which is already in distinct populations, so the results turned out less interesting, which is like, I had 3 populations I pulled out, and I got like 3 very nice, neat clusters. So it wasn't quite as cool as the paper, but I was able to reproduce it, and I was able to do it myself. And this was actually the first time I ever used Python. So this was a fun project. I recommend doing something like this. You can probably find better SNP data available these days, including from the 1000 Genomes project or any other source you can find where you just need SNP data along with location data so that you can check whether the final PCA plot ends up looking like ends up being related to the populations people are in or where they live. Mostly where they come from ancestrally, right? Because that's what matters for how their DNA turned out to look, or how their DNA turned out to be more or less similar to other people. Okay, so those were some specific ideas. Now, I have one additional idea that I separated out because it's very software engineering related. So for those who are inclined to get particularly into the software engineering aspects of bioinformatics, I would encourage you to pick a bioinformatics tool and make it easier to use. So there are many bioinformatics tools that are, or there are some bioinformatics tools that still have a team behind them, that are still very well maintained, that have people answering issues on GitHub and rotations and stuff like that. I work on one such team that's managing this for deep variant, but there are others, it's the same thing for many other tools. IGV is another example where you have a community still supporting this, and it's not just, left out in the cold. But many academic tools are a little bit left out in the cold and a little unmaintained as the person who was maintaining them moves on. I have a few tools that I try to maintain myself, but it is a little bit of a struggle, right? I am not often doing this kind of analysis a lot anymore. I don't get new data of the same type, so I don't use the tool myself all the time. And so it can be a little harder for me to maintain it. And since I wrote many of these tools before I knew like really good software development practices, there's no continuous integration or deployment or testing. I have some tests actually for some of them, but they don't run automatically when a new pull request comes in or anything like that, which is totally what we do for like real software engineering projects that I'm part of building these days. So these older tools, they're a little bit like, I'm scared to change things. So anyway, there are also many tools that have just been completely left behind. Some tools are not very easy to install. So here comes like the ideas. Many bioinformatics tools are hard to install. We try to make it easy, but it's not always easy. With some of the newer tools available, you might be able to make this easier. So one thing you could do is figure out how to install a tool, wrap it in a Docker container, like get that working to a point where you can take the Docker container and just like run it straight from a new virtual machine on the cloud or something. And that Docker container should like work out of the box. If you can do that, you can take a bioinformatics tool that is like really hard to run and potentially make it much easier to run. I have a colleague who did this with CNVnator, which has been super helpful because, you know, it makes it easier for other people to use the tool now. So that's great. Another thing you can do is look for bioinformatics tools that are still using Python 2 and convert them to Python 3. All these things are a little bit dangerous. Another thing, so try to get it working on your own first. Make sure that you have specific test cases and so on. Oh, I'm low on battery. And the third thing you can do to help everyone when it comes to bioinformatics software is to improve the documentation. Have some friendly tutorial code or examples or something. If there aren't good examples with test data and stuff like that, like a small test set, like we'd like to have like a quick start or something that anyone can run really quickly to make sure that the tool works as expected. They can see what the output looks like. They can get an idea of whether it'll work for their data. You could add that to the documentation of existing tools that might not already have it. So one thing when you're contributing to other people's code is, of course, try to get it working on your own first. When you do have something that you're pretty confident is a good actual improvement, then contact the developer or file an issue on GitHub to at least ask, or you can file a pull request. But I think it helps to just reach out to them and say, hey, I want to help. I've tried, like, I love your tool. I'm trying to make this change, and I think I got it working. And then you really want to make sure that you have some tests and some like very specific ways for them to verify that the work you have done did not break their entire software and is actually functional and is working. So that's a great skill anyway, testing your software, making sure that you end up with a good change that is actually doing what it should be doing, and that you can document that work to make sure that you can also show other people that it's working, not just that you can tell that it's working because, well, I ran these tests. No, show them the tests, show them how they can reproduce the tests, or even add a little bit of automated testing if their software doesn't have it already. That could also be a good contribution in and of itself. And this is a great thing to learn for general software engineering because it's super important, and it's what makes our software more maintainable in the long run and makes it so that future developers can contribute more easily. So that's what I would say if you're going to do software engineering related projects. The documentation is easier. You don't have to worry about breaking their tool if all you're doing is adding a bit of documentation and some test cases that users can run through. So that's always a good place to start. And for the Docker container as well, you also don't have to do that. You can just, host a Docker container somewhere and then tweet about it. And you can add a comment here saying that you did that for a certain tool and show how people can use it. and document how to use your Docker container and stuff, right? Because we can't guess that. So this gives you hopefully a lot of ideas for how to get started with some concrete bioinformatics projects. You will, of course, have to pick the papers yourself. I can't really do that for you. I think the genes mirror geography within Euro paper was fantastic, but it's often hard to find any that are quite that crisp. Usually there's a lot going on in these papers. So you can also just do a particular analysis and reproduce that for one figure or something. It doesn't have to be the whole paper. But hopefully this gives you some ideas on ways you can get started with a specific project in bioinformatics. So leave a comment below if you want to share a particular starter project that you are working on, or cool papers that you think would be nice to reproduce, or particular kinds of bioinformatics software that is hard to run and you wish that someone, maybe not you, who read the comments, could wrap it in a nice Docker container or help make it have better documentation or something and make it easier to use. So you can also ask each other for help in the comments. And I will link the Twitter thread in the description of this video, so check that out. And that's it for this video, so thanks, and see you next time. If you haven't already, subscribe to the channel. YouTube thinks you'll like this video, and this is a video that I think you'll like. Engage with the YouTube video in the way that you see fit.